My name is Pete Knapp, and these are the results I got from the TBS in Bristol, where I produced pairs of droplets and collided them together with different speeds and angles to see how their outcomes differed. So collision outcomes are important because of atmospheric science and drug delivery, spray drying, and so on. And droplets around 40 microns were produced, which is around 10 times smaller than previous studies. The stroboscopic images were used here, so each subsequent frame is actually two different pairs of droplets, as seen from the slight wiggle in the video. This one is a coalescence, and there are 5 different outcomes, as shown in the next slide. We have 2 droplets coming in, one in red and one in blue. And the first outcome is when they simply bounce apart from each other. The second one is a slow coalescence where they, without significant deformation, produce one droplet that's well mixed. The third one is a fast coalescence where they come in and kind of pass through each other but then bounce back again to form a well mixed droplet. And then we have reflexive separation where they do much the same thing but then separate, but they're still fairly well mixed and they can produce more than 2 secondary droplets. And the third one is a stretching separation where they don't well mix at all, but they kind of graze past each other, but they sometimes produce well mixed secondary droplets. I imaged the droplets as they approached and then used Python to track the droplets. I used that to work out the speed and the angle that they approached, and I repeated this with different speeds and angles and also with different viscosities by colliding water with water and also sucrose with sucrose, so there were 2 different regime maps that I was able to produce. The first one shown here was the 2 water collision droplets, and the different colors show the different outcomes. So in the blue squares are the stretching separation, the green triangles are coalescence, the black circles are reflexive separation, and then there are these yellow ones which were kind of a mixture of 2. Because there was a wiggle in the video, sometimes you got this kind of mixed outcome. You can see that there are 3 different lines here. They represent the 3 different models, and all 3 are pretty close to the data. With the sucrose droplets, the error bars were much larger, and that's because the droplets were moving around a lot, because over time the dispensers kind of crystallized, and then the trajectory of the droplets was moving around a bit over time. If this were to be repeated, then that could be done with a more stable droplet, and hopefully the error bars would be smaller. And overall, all the 3 models fit pretty well, although the Sommerfeld and Pasternak one seems to fit the data a little less well, and that's shown in the solid black line.